All right, so the last big part that we have for this unit is principles of development. So principles of development, there are two thoughts on this. One is a very old thought that doesn't have any science behind it. One is a thought based on scientific data. So I like to share both because I think it's interesting to see how people once viewed reproduction. So epigenesis versus preformation. So epigenesis is a theory that we now hold that embryos develop from a mass or a group of undifferentiated egg cells. Essentially, you could say that these are growing from stem cells. Stem cells are cells that have all the genetic material. They're ready to go. You just haven't told it which part of the genetic material it needs to follow, because it doesn't need to be all the different types of cells. It either needs to be a liver cell, or a brain cell, or a bone cell, or a nerve cell, or a pancreas cell, or a stomach cell, or a muscle cell. It needs to be one of those. It can't be all of them, but it hasn't been told what to do yet. So as these cells are splitting and dividing, they're simply just doing that. They're splitting and dividing. They're not becoming specific types of cells. And we see this. There's data, tons of data that backs this up. And we see that our first early steps beyond zygote, when we start developing into an embryo, we are undifferentiated cells. It's not until after a process called gastrulation that our cells start to differentiate into things like external cells, inside cells, and cells that are in between, and eventually become our skin or our bones or our organs. So they're undifferentiated to start off with. The old idea, then, you're probably thinking, then what do we think that we used to be? The old idea, get ready for it. We thought that you were basically a sperm or an egg was literally a miniature version of yourself. So, and this is kind of what they thought they looked like. This is taken from a textbook, from a journal, scientific journal, February 7th of 1695. That, my friends, is a sperm, and those two things are also sperm. This is what we thought sperm anatomy looked like prior to the invention of the microscope, or just common sense. So yeah, that's preformation, and as you can imagine, that is not the idea that we hold anymore. So epigenesis is definitely something that we like to study and go in and see what's happening, because it's really cool. There's a lot of big studies that have gone on with epigenesis, looking into human development and the right environmental cues that are given, how much of those cues are given, and when they're given in the developmental process that can make a difference in how systemics develop, how your systems develop, how you develop, and what makes you you. So it's a really cool thing to look at. So fertilization and conception, there's a difference between the two. So conception is simply the process of the sperm entering the egg. There's a whole act that goes along with that, because I used to tell my students that, hey, congratulations, you were the fastest swimmer, meaning that you were the first sperm to have met the egg. We now know that is not true. It takes the first 1, 2, 3, or sometimes even 4 sperm, or sometimes more than that, to cause a chemical reaction that changes the coating on the egg, that will then allow the next sperm to enter. So the first couple of sperm to reach it are simply kind of like sacrifices. They chemically alter the egg to allow preceding sperm to enter. So you weren't even the fastest swimmer. You're like 4th place, but you know what? You're the one that made it through, so congratulations. But that's just the process of getting into the egg. She's got to lower her shields first before she lets anybody in. Fertilization is simply when the fusion, once the sperm gets into the egg, it's the fusion of the 2 nuclei. For humans, it would be the haploid nuclei from the sperm and the haploid nuclei from the egg fusing together to form a diploid nuclei, which would then start to develop. So yeah, there's the process of fertilization when the 2 genomes are connected together. Whenever a egg cell and a sperm cell fertilize each other, before any division starts, that resulting fertilized egg is called a zygote. Once it starts to divide, it is called an embryo, and that is the difference between a zygote and an embryo. Fertilized cell that hasn't divided yet, zygote. As soon as it divides into 2, embryo, all right? When the embryo starts to divide, it forms kind of like a division at around it, and we call that a cleavage furrow, and it forms a cleavage plate, which then splits those in one cells into two. Remember from mitosis and meiosis, the process ends with cytokinesis, the splitting of the two cells, or the goo inside the two cells, right? So, and then it cleaves into to become two separate cells. So whenever we start talking about embryos, this is where we start talking about cleavage. And there are multiple different patterns of cleavage that we see in the animal kingdom. And it's wild how the different ways that this embryo will split itself up when it starts to divide. It's so big that I'm not going to cover it. It's actually a huge section of the book, and it's very complicated. It's neat to look at. If you ever get really interested in a specific organism, I would definitely say checking out its cleavage patterns. But for the purpose of this course, I'm not going to actually cover that information. But know that it is in the book if you want to see it. All right. So once the embryo starts to divide, for a good while, it's essentially just a solid mass of undifferentiated cells. The cells go all the way through, all over the place. If you were to cut it open, there would just be cells everywhere. All right, so we call that a morula. It's a solid ball of cells that are dividing. They're still undifferentiated, right? But in order for us to start having differences between the cells, we need cells on the outside, we need cells on the inside, and we need cells in the middle. We've got to kind of like have a point of origin, a point of reference to start dividing them. So what starts to happen, there's our solid ball of cells. What starts to happen is the next step after a morula is called a blastocyst. And for a blastocyst, the cell are starting to push towards the outside and hollow out the inside of this ball. So a blastula is a hollow ball of cells. There's a blastula. You can see now we have cells that cover the outside. We can have cells that are going to be on the inside, and we can even have cells that are in the middle. And so we start getting different layers of cells, which are going to develop into the different types of tissue, which will become the different types of organs. This is all how the process of development occurs. This is all a process called gastrulation. So know what an embryo and a zygote is and how they're different. Know the difference between fertilization and conception. Definitely know the differences between when cleavage starts, and the difference between a morula and a blastula. So here you can see, this is just human reproduction. This is, here we have an ovary and a fallopian tube and part of the uterus. And you can see that the egg is being released. Fertilization usually occurs in, conception usually occurs early on, as soon as the egg has been released from the ovary into the fallopian tube. You have the process of conception, then and also fertilization will occur around this area, and then that zygote starts to develop a cleavage furrow, and as soon as it splits into two separate cells, it is now an embryo. Cleavage will continue, and as long as it's a solid ball of cells, it's called a morula. Once that morula starts to hollow out on the inside, it is now a blastocyst. And if the uterus during the menstruation cycle, when progesterone levels increases and the lining of the uterus thickens and softens, if a fertilized egg then attaches to the wall, releasing a chemical cue, then the progesterone levels stay high in the female's body, and she'll start going through the early symptoms of pregnancy. If an egg is released and it is not fertilized, it generally will not stick to the inner line of the wall. Because it doesn't stick, no chemicals are released, progesterone levels decrease in the female's body, and the inner lining of the uterus simply refreshes itself to get ready for the next egg that's going to come through. And of course, I just described the process of menstruation. Very sciencey sounding, ladies. I know what happens. I personally don't know what happens, but I hear it's pretty bad, and that it's a very big roller coaster, and that's because it's dealing with hormones, and hormones can really have a big impact on our physiology and our biology. But this is the process of ovulation and menstruation. So boom. So let's take a moment, let's talk about sperm anatomy. Remember earlier when I said that in order for the sperms to get through the egg, the first couple of ones must attach to the outside and cause a chemical change to the egg? That's exactly what happens. The top of the sperm, there's a capsule called the acrosome. The acrosome is filled with digestive enzymes. So here's the anatomy of a sperm. You can see the tail. Here you can see the base of the tail. This is where all of the mitochondria are, and they're producing all the power that gets the flagella to whip back and forth. And that's also why all of the mitochondria you have are from your mother, because this part of the tail will never enter the egg. Therefore, you get all of your mitochondria from your mother. But at the front of this is this, on the head of the sperm, you have the genetic material inside, but there's also this digestive enzyme in the area called the acrosome. Once that acrosome, once the sperm reaches the egg, the first few that get there, the acrosome pops and the digestive enzymes start breaking down essentially this force field that surrounds the egg. Once they start to get through and a sperm finally makes it through, it ejects the genetic information. Again, the tail with the mitochondria never enter in. Once this piercing happens, then genetic material from one sperm is inside of the egg. You see these little groupings, they're called cortical granules. Once this change happens, a chemical is released, it causes the cortical granule sacs to rupture, and when they do, they essentially reinforce the shield around the egg, stopping any other sperm from entering, and that's what we want to happen. You don't want 3 or 4 copies of dad's genetic material inside the egg. It would spell disaster for the egg, and the egg is simply, it may develop a little bit, but it would stop early on, and it wouldn't result in a viable offspring. Just 1 copy of dad's, well, half, right? Well, just 1 full copy, so just a haploid amount of information coming in to mix with a haploid that mom already has in her egg, combined together to make it diploid. No more, right? No less. But this is essentially the process of conception. So that's what we just kind of saw here. Again, in order for the egg to get the sperm on the inside, the sperm's acrosomes have to pop, have to digest and break down her outer lining. As soon as one of the sperm gets in and releases the genetic material, the granules pop, reinforce the outer lining so nobody else can get in, and then we can see the process of fertilization occur, where the egg and sperm genetic material actually combine together.